people by email, and it doesn't mean I will answer the email right away. All right, so today what we're going to do is we're going to finish plasmids. And remember, the whole discussion around plasmids is for you to be able to visualize this idea of a mechanism using conjugation of natural plasmids that allows for the exchange of genetic materials. So some plasmids are not they are not conjugative. They can't do the cell to cell, but they still may be able to exchange genetic information with the chromosome. But then we have the conjugative plasmids, which have that ability to go from cell to cell and bring with it characteristics. So I introduced you to the fertility factor last week as the example plasmid. It's found in a lot of different cell types. Are plasmids, and there are a lot of them. So those are the resistance plasmids and some of the different bacteria. They come in mostly amongst the Enterobacteriaceae. And again, the important point is not remembering each one of the names. But if you just come over to this side and realize all the different types of antibiotic resistances these are plasmids carry. And these are conjugative plasmids, which means they can move from Shigella to E. coli, for example, or Salmonella to E. coli. So therefore, it's a really good way for them to exchange genetic material and gives you a vision of why antibiotic resistances become highly problematic. Because if we have a resistance plasmid in one cell type, and they come in contact with a population that can accept that conjugative plasmid, then that group of bacteria can suddenly become antibiotic resistant as well. We have the coli plasmids, which we'll talk about a little bit more, as we will the R plasmids, which actually encode colicins. So colicins are bacteriocins. What are bacteriocins? Does anybody remember? I'm going to do this again, but what is a bacteriocin? Right, so it's usually proteinous material that's secreted by one bacterial cell that will kill other bacterial cells. These also can be conjugative. Then we have what's called the virulence plasmids, which carry virulence factors. We'll go in a lot more detail about the ENT, the K88, and the TI plasmids. Again, they carry certain types of virulence factors that therefore make the bacteria even more pathogenic. Remembering our definition of virulence, virulence factors don't in and of themselves make an organism pathogenic. They have to be pathogenic, but if they have virulence factors, that increases their virulence or pathogenicity of that organism. We'll talk in more detail about these three specifically. These two are especially important because they differentiate two major groups of E. coli that cause serious problems in terms of various types of infections. And then we can have the metabolic plasmids. And we're going to talk about the NIF-NOD and the DIG plasmids. So the SIP plasmid is also called the NIF-NOD plasmid. And then these plasmids that are found in pseudomonas are about organic removal of organic materials, such as the purity of grape oil. So we as like oil spill. So we're going to talk about some of these different classes in a little more detail. And one of the things I want you to keep your eyes open to see is that a lot of these plasmids also have transposons built into them. We're going to talk about transposons in a little while. So let's talk about the T plasmid, right, or the TI plasmid. So this is a metabolic, this is a virulence plasmid. So TI plasmids are found in Agrobacterium tumefactions. Its name tumefactions would lead you to believe it is involved in tumors. These are classic plasmids that are present in bacteria that have a way to actually infect plants and cause the big tumors you see on plants like the crown galls you see on some specific plants. So the way in which this works is Agrobacterium has an ability, by the way, you can actually transfect plants with Agrobacterium. So Agrobacterium grows in the soil, and basically what can happen is if there's some kind of wound that usually occurs in the lower stem, then that bacteria can gain access to that tissue, and it does so not by infecting, but rather it actually injects a small amount of tDNA that's found in the TI plasmid. So this is the bacteria. This is its chromosome. So it's not like this entire bacteria infects, but rather it actually injects this small amount of tDNA, and that tDNA gets integrated into the chromosomal DNA of the host. That tDNA is considered a virulence factor because that then causes the plant to start producing these huge crown galls that we see often at the base of plants. So the TI plasmid then is a way for the agrobacterium to inject part of its DNA specifically from the plasmid, and increases the ability then of this bacteria to actually carry out an infection. So without the injection of this tumor-inducing DNA, that infection would not be possible. So the agrobacterium is infecting, but it's the tDNA that's causing that huge tumor. So from a perspective of the health of plants, obviously that's still deleterious. But from the point of view of a molecular biologist interested in adding genes to plants, to genetically modify plants, this becomes a very useful system. So we know that the T region of a TI plasmid can actually be placed into the genome of a plant. So if we take a TI plasmid with its T region and add a cloning site, we can actually insert DNA. And in so doing, we can make a transgenic plant. And when you insert the gene of interest into this tDNA region, you can actually inactivate it. So it won't cause the formation of a tumor anymore. But it still will take that DNA into the plant's DNA. So we have a perfect vehicle to get our gene of interest to go from the plasmid and get integrated into the chromosomal DNA. But in this case, the plant will not produce a tumor, but rather will express the gene of interest. So this use of the tumor fascions to actually modify plants is a common way to make transgenic plants. So a classic example of what you might be interested in doing is perhaps you're interested in adding a herbicide. So, adding a herbicide resistance gene, so that when you treat your field with herbicides to get rid of the weeds, your plant of interest will not be killed. So again, we take the DNA region, the tDNA region, we insert our gene of interest. So this is our herbicide resistance gene. The region gets integrated. So here's the tumor fascions. This is agrobacterium, to which we've added the tDNA. We then carry out an infection by adding fascions onto our plant. And then once we actually do that and we look at the leaf disc in the presence of herbicides, we see that certain of the cells have the capability of growing in the presence of the herbicide, because we've added herbicide resistance genes. So many of the ways of making transgenic plants is making use of tDNAs and making mutants. So you may read about a tDNA insertion library. It's all about adding tDNA to disrupt genes or add genes for the purposes of studying either gene expression or adding a functionality to a plant. And of course, the world of agriculture is full of attempts to modify plants to allow them to resist cold, to resist drought, to resist insects, to resist other things that result in lower yields of plants. In fact, the major plant product, does anybody know what the major plant product in Canada is? Something that we export the greatest amount of, one of them? Anybody? Okay, we do make ice wine, that's true, but more in terms of plantings, yeah, canola oil. So soybeans, and they're almost all genetically modified. If you ever take a drive out in the country and you pass all these fields with little pioneer hybrid signs in front of the fields. They might be cornfields. Most of our corn is genetically modified as well, and they're owned by Pioneer Hybrid, the line. So many of the plant products that you're consuming on a regular basis have in fact been genetically modified to increase crop yield. All right, so that's an example of a metabolic plasmid. Now let's turn our attention to the colicins, or the bacteriocins. So just a reminder about what bacteriocins are. There are a variety of different small molecules. So the names of them, again, are not important. So here's two examples that are found in that target gram-positives. Here's some that target gram-negatives and the way in which they do it. So first of all, the reason I'm showing this is, again, to sort of get you to think about the difference between a gram-positive and a gram-negative cell wall. So again, a gram-positive has a thick peptidoglycan layer, a periplasmic space, and then the plasma membrane. So these bacteriocins can move through relatively easily the peptidoglycan layer, and then they impact onto the cell membrane and usually cause some kind of alteration. So they may inhibit peptidoglycan synthesis itself, because as we'll talk about, peptidoglycan is made in the periplasmic space. They may cause pore formation to allow for leakiness, or they may actually enter the cell. Same scenario with the gram-negative, only in this case, notice what these bacteriocins have to do. They have to go through the outer membrane and an inner membrane. So that means we need proteins in the outer membrane and we need proteins in the inner membrane to allow these bacteriocins access. So we need outer membrane proteins, and then we need, and the same scenario can happen where we can have pores form, and then the functionality of the bacteriocins can be anywhere from causing leakiness to inhibiting DNA gyrases or RNA polymerases. So DNA gyrases are involved in what? Why would if I inhibit a DNA gyrase, why would that inhibit the growth of the organism to which I'm inhibiting DNA gyrase? What does DNA gyrase do? Yes, so DNA gyrase is about supercoiling. Removing supercoils, adding supercoils. So the DNA will not function correctly. If I inhibit RNA polymerases, obviously I'll inhibit RNA synthesis. So the bacteriocins have multiple effects. And for our point of view, I'm not interested in going into detail about all the different types. I want you to just visualize that bacteria secrete molecules that then gain entry and cause pores to form in other bacteria. And sometimes they deliver a particular target into that organism. So if all it's doing is causing a pore formation, then that just causes a leaky membrane and things spill out. But they also can actually be molecules that target specific function within the cell, causing the death of the bacteria that they're targeting. So those bacteriocins that I've just shown you on this slide can be encoded, as I said, by plasmids. So the plasmids themselves may encode the bacteriocins. So in E. coli we have a number of toxins that are proteinous that are encoded on these coli plasmids. And not only can they target E. coli, generally they target within the Enterobacteriaceae, which includes the bacteria that are in your gut, anywhere from E. coli to Enterobacter, Gala, pseudomonas, etc. Okay, so again, remembering again that these coli plasmids are conjugate. So if one bacteria has these bacteriocins, they can pass them on to another, and that could mean that they gain a functionality in terms of being able to destroy the surrounding bacterial population. Some examples of metabolic plasmids. One is the D plasmid or the big plasmid. So we go back to our chart again. Okay, what we're really talking about is these plasmids in this region that specifically are going to break down what we would define as toxic organic compounds. So these plasmids have various names. They're generally found in pseudomonas, and if you look at that category, it was all pseudomonas that carried them. Pseudomonas is a psychrophile towards a psychotroph, so it likes cold temperatures, but not really, really cold temperatures. What these plasmids do, again, is confer ability to degrade substances. Now, why are the Pseudomonas carrying them? Well, it allows them to gain nutrients from material that most other organisms can't, including things like breaking down petroleum, pesticides, and herbicides like Agent Orange and jet fuel. But of course, the advantage is that they can then be used to clean up sites that are contaminated with these various organic materials that are very hard to actually remove. So they can even work at ocean temperatures that are 4 degrees Celsius to help break down the oil slicks that develop in an ocean. Does anybody know what Agent Orange is? It is a herbicide. So it was used in Vietnam to defoliate the forest so that the Americans can see Vietnamese. And it's a highly toxic. At the time, it was not clear how toxic, by the way. It was a classic herbicide, but now, of course, determined it's a highly toxic herbicide. All right. So another example of metabolic plasmid is the NICNOG, or the bioc17 plasmid. This is found in rhizobium. So again, this is the bacteria that forms symbiotic relationships with the root hairs in a plant. So if you've ever pulled up, especially leguminous plants, so there's a plant under the soil, you have roots, and then you have root hairs, and looked at the root hairs, you see these nodules on the root hairs. And the nodules are actually an interaction of the bacteria and the plant. They live in the nodules of the plant, and the bacteria carry out the nitrogen fixation. So it's the bacteria, i.e. the plasmids in the bacteria, that have the non-genes and the nitrogen genes that allow nitrogen to be broken down, like nitrogenases. And the non-genes are involved in the nodule formation. And the nodule formation is important because in order to carry this out, we have to create an anaerobic environment. So the entire bacterium enters the plant cell, and there are different species of rhizobium that attach to different species of plant cells. So in other words, if I'm bacteria X, I will only infect a particular leguminous plant based on the receptors that are present on the root hairs of the plant. And then this bacteria sort of can interact specifically with the receptors. So it's not like all of the rhizobium species interact with all the leguminous plants. They form a very specific species-to-species interaction amongst the leguminous plants and rhizobium. And basically, it is an infection through the root hairs, and it's due to the receptors, and the receptors have lectins. The receptors have carbohydrates on them, which is true really of any external receptor on a eukaryotic cell, and the capsule around the rhizobium have lectins. So lectins are a molecule that will actually bind to carbohydrates. So they have a specific interaction. So the specific lectin on the capsule of the rhizobium interacts with the specific receptor and the glycosylation on that receptor in the plant. In order to fix nitrogen, by the way, we need an activated oxygen. So the reason we form these nodules and they form them within the plant is because the lake hemoglobin that is present in the plant can bind oxygen in the root nodule, which results in creating low oxygen within that confined area. And then that allows the nitrogen fixation to produce nitrogen, starting with ammonia, in this kind of symbiotic relationship. And this is a true endosymbiotic relationship because the bacteria is living inside the actual plant. All right, so we dealt with metabolic plasmids. We dealt with Fanconi plasmids. Now let's talk about virulence and pathogenicity plasmids. So these again are going to be plasmids that are carrying genes that are going to cause pathogenicity or increase virulence. And they are also found in many different bacteria, and again, they are conjugates. So let's go over our terms again. Pathogenicity is the ability to inflict damage, which can result in a disease. Pathogen is the organism that's capable of causing that damage. So a disease is any change in state of health leading to abnormal function, and virulence is the degree or intensity of pathogenicity. The problem you will run into is that people start to use virulence and pathogenicity as interchangeable terms. They are not interchangeable terms. The organism can make all the virulence factors in the world. If it is not a pathogen, it has no ability to attach to its host. It doesn't matter if it secretes all sorts of other things. Fungi, for example, secrete multitudes of proteases and lipases, etc., into their environment. Theoretically, those are virulence factors, but most fungi are not pathogens, so they don't have any effect on our cells. So those enzymes would only be useful to a pathogen. The fungi uses those enzymes simply to collect food because they're heterotrophic. They send out those enzymes, they break down their environment, they bring in the subunits. So a virulence factor then is a factor that contributes to the virulence of a pathogen. So you can think of pathogenicity factors as things causing direct damage that cause it to be a pathogen, and virulence factors aid in making something more pathogenic. So some of the things factors that can contribute to pathogenicity, those that cause direct damage, such as the production of toxins that directly damage cell, as well as degradative enzymes like proteases, lipases, and DNases. So later in term in the lab, you'll evaluate some of these classic enzymes. We'll look at the ability of some bacteria to produce DNases and secrete them, which can break down DNA, secrete gelatinases that can break down gelatin, which we find in connective tissue, as well as enzymes that can destroy white blood cells, etc. So when you get to that point, it'll relate back to our discussion of things that cause direct damage to a cell. Things that contribute to virulence generally affect pathogenicity indirectly. So these things that cause direct damage can generally contribute directly to pathogenicity. But again, in my example, if I secrete enzymes, that doesn't make me a pathogen. I still need to be able to infect that particular cell in order to be a pathogen. Some of those things that we might say indirectly affect pathogenicity would be things like the presence of an antibiotic resistance gene. So a bacteria that's making antibiotic resistance genes is not any more pathogenic than any other bacteria. But what it has is the ability to survive when antibiotics are used to try and kill them. So that increases pathogenicity indirectly because they in essence are able to survive. So also examples are the presence of sticky fimbriae, which allow things to attach. So remember, if you are a pathogen, it is in your great advantage to be able to stick to surfaces. So things that make you stick make you more capable of infecting. So things like fimbriae, things like pili, things like capsules. So if you remember last week when we talked about natural transformation and the difference between having a capsule for Streptococcus and not, the one that didn't have the capsule was not a pathogen. Okay, so the presence of the capsule can define the ability to increase the ability to infect. So capsules and certain cell wall components then allow, this is a capsule, classic capsule, allow the pathogen to adhere and also protect the pathogen because the capsule prevents the movement of material into the cell. It also prevents the cell from becoming dehydrated. So all these things become important. Capsules prevent things like antibiotics from getting in. So within these virulence factors, we can have two major classes of toxins. And we're going to come back to these several times throughout the course because they're important concepts. So the names exotoxins, so exo means to be outside, so these are secreted enzymes by a bacterial cell. They are proteins, they're soluble and they're heat labile, so we can heat them and destroy them. They're secreted as the bacteria grows and they target the host. So then we can have neurotoxins, enterotoxins, and cytotoxins. So the neurotoxins are specific to nerve tissue. These are examples. Enterotoxin affect the intestinal mucosa. A more general term would be cytotoxins that affect many tissues. Many of these do is disrupt host plasma membranes, causing cell lysis, and some of them just block function. So we're going to talk about a variety of exotoxins, especially the AB exotoxins, like cholera and botulism, which are two examples of diseases caused by AB exotoxins. The endotoxins are a little different. So these are not things that are released from a bacterial cell. They are, in fact, part of the bacterial cell. So endotoxins are actually not normally released unless the cell becomes damaged and lysis. They're not proteins. They're actually lipopolysaccharide, or LPS. And specifically, they are lipopolysaccharide that is found on gram-negative cell walls. And we'll talk about this at length when we start talking about cell walls, because there's a component of the lipopolysaccharides that is an endotoxin. So oftentimes, when you eat contaminated food, it may not be a secreted exotoxin that makes you sick, but rather the dead bacteria that end up in your gut, because they don't survive your gut, but in the process, because they start to lyse, they release endotoxins, and it's the endotoxic response your body actually responds to. So we'll talk about that going forward in several lectures. Okay, so let's talk about a couple of examples of virulence of pathogens in plasmids, and I'm going to concentrate on the N plasmids and the K88 plasmids to give you an example of the kinds of characteristics these plasmids can lead to. Both of them are commonly found in pathogenic E. coli strains. The N plasmids, and remember both are conjugate type plasmids, so the N plasmids actually encode exotoxins. So remember, exotoxins are secreted proteins that affect the functionality of the host cell. And what these exotoxins do is they cause lesions or alter the permeability and cause hemorrhage in the epithelial lining of the intestine. This is classic bloody diarrhea. So when you get a bacterial infection from E. coli, these exotoxins are released, and basically they cause your intestines, the mucosal layer, to start hemorrhaging, and it leads not to diarrhea, but actually bloody diarrhea. So not only do you have diarrhea, but there's blood in amongst the liquid. Bacteria the E. coli that actually carry these plasmids are called enterotoxigenic bacteria. They cause severe problems, especially in the young. And why? Or those that are more frail. Why? Because diarrhea results in severe loss of fluids, and so somebody can die very quickly if they actually haven't gotten fluid replacement therapy very quickly. The K88 plasmids are carried also by E. coli strains, and these actually encode adhesive fimbriae. So fimbriae are pili that are found all around the surface of bacterial cells that, as I have already indicated you, increase the ability to attach. So basically these allow adherence. And once they can adhere to the lining of the gut, they can invade. So the presence of these specific pili increase the ability of E. coli actually to invade into the epithelial lining of your gut. In this case, there's no invasion. You're just causing the gut to basically stop functioning correctly. In this scenario, there's actually an invasion where they attach to the gut and they actually invade. And they can induce a very severe endotoxic response. Remember, that's a response to part of the gram cell